So today we will be covering the need for normalization step in single cell RNA-seq data. So in the figure they have shown the clustering of the macrophage population from study in human and mouse lung cancer. And I have given the reference to the paper and also the blog from where I have taken these images. So in the left figure we can easily see the total count is higher at the periphery as compared to the cells which are at the center part. And in the right hand side figure, coloring is done by the expression level of this particular gene S100A9 gene, and the product of this gene is basically important in the inflammatory response. So it is easy to see that this particular gene, the raw value is highly correlated to the total count because the center of the both plots have low value in count and expression, and the periphery area has the higher expression and the higher counts. So one conclusion we can make from this is the number of this gene transcript will goes up when the total transcript capture in a cell will go up. But this is not obviously the true. So for any single cell data where the total counts, that is the sequencing depth, shows these kind of variation. So their normalization is must. So in the figure, we can see how the single cell RNA-seq normalization changes the expression pattern of this particular gene. So it seems to be there is no correlation between the expression of this particular gene with the total count, because the expression profile here is simply indicates that at the center the expression of this particular gene is higher compared with the cells which are at the periphery part. We can say that the variance of this particular gene expression is not dependent on the technical variation, that is the sequencing depth, and the differences we can see here is because of the true biological factors, not because of the technical noise. The next question is, how does single cell RNA-seq normalization work? So the normalization step is divided into two steps, that is scaling and transformation for simplicity. And Surath also apply a global scaling normalization method that we call log normalize. So first, log normalize method is divided into two steps, step 1 and step 2. So first we apply a size factor to scale the data in the first step. And this step 1 is basically turns the absolute count into concentration. It means like how many transcript of gene A are there in per cell. So in this we are calculating the feature count for each cells, and then we are dividing with the total counts for that cell, and then multiplying with a scaling factor, that is 10 to the power 4, which is a default one in Surath pipeline. And then this value is transformed in the next step, that is the transformation step, that is step 2. So here we are doing the transformation that will give the normalized count for gene A for this particular part. So this is log transform, and the transformation is done to reduce the skewness in the distribution and also to further regress the technical variation. And the simplest type of transformation is log transformation that we see in Surath pipeline for the single cell RNA-seq data. And the one important point here to note that is why we are adding +1 in the log transformation, why we are not simply doing log transformation of X. So the reason is, like, instead of using log transformation X, so we are adding +1 to avoid the zero counts, because we know that the zero counts are common in single cell RNA-seq data. So without adding +1 in the normalized value, so the log transformation value will hit error, because when you do the log of the zero value, so it will give you the error. So that's why we are adding +1.